Hello students, so once again I welcome you on our YouTube channel, Little Kingdom School. So we are discussing the chapter 1, Sexual Reproduction in Flowering Plant of Standard 12. So keep your notebook, textbook and pen with you, which is going to help you to understand this topic much better if you are preparing notes. Okay, so first of all we are starting with the revision of what is female reproductive part, because this particular topic we discussed in the previous video. So let's quickly revise the topic. Female reproductive part in flower is called pistil. Done. It's look like this. This basal swollen part is ovary. This center part is style and this portion is known as stigma. As we know, stigma provide landing site for pollen grain. Style connect stigma and ovary. Clear? And ovary is a basal swollen part of pistil which contain megasporogenesis. Megasporogenesis, that means ovule. If you remember, we draw a diagram like this in a previous video. So this one is ovarian wall, which I draw here for a reference point. This one is placenta, nutritional tissue, which provide nutrition to the ovule. So this whole structure is known as ovule. The stalk is known as funicle. The junction point between the stalk and the body of ovule is called hilum. Inside, in this structure, the basic tissue present known as nucellus in this portion, known as nucellus. And in the center of nucellus, the most important structure for sexual reproduction is present, called embryo sac. Embryo sac. As this embryo sac is the most important for a sexual reproduction, it need to protect it. That's why this layer or this membrane provide protection to developing embryo sac. So this one is outer integuments, this one is inner integuments. So in last video we discussed about the structure of ovule, and yes, one more thing, the end without this integuments is called micropyle end, and end with integument is called chalazal end. So in today's video we are going to start the topic called Megasporogenesis. It's a very similar to microsporogenesis. What happened in microsporogenesis? Microspore produced. And what happened in megasporogenesis? Megaspore produced. Clear? So we are going to discuss megasporogenesis, but for that you need to remember this portion of nucellus and embryo sac. So let's start with the next topic called Megasporogenesis. Megasporogenesis. Clear? As we know, we are using mega word to indicate the female reproductive structure, and we are using micro word to indicate male reproductive structure. Right now we are talking about the female reproductive structure, so we are using megasporogenesis. Megaspore, a structure which is going to develop female gamete in future, and genesis, that means production. So the process of production of megaspore is called megasporogenesis. So let's start. Now as we know, what happened or what is the structure present inside the ovule? nucellus tissue which present surrounding the embryo sac. So now I am drawing only this portion, embryo, not embryo sac, nucellus and embryo sac. Clear? So let's start. So suppose this one is nucellus. Clear? It is a group of cell, thousands of cell present in nucellus tissue. These all are cell. All the cell of nucellus are diploid. We know that whole plant is diploid. Each and every structure of plant is diploid. That means root is diploid, leaf is diploid, all the branches are diploid. Done. Flower is also diploid. Male reproductive part is also diploid. Female reproductive part is also diploid. When we said the whole female reproductive part, pistil, is diploid, that means stigma is also diploid, style is also diploid, and ovary is also diploid. That means the structure present inside the ovary is also diploid. The whole ovule is diploid, and if the ovule is diploid, that means the cell of ovule or the tissue of ovule, nucellus, is also diploid. Clear? So the each and every cell of nucellus are diploid. What happened next? Any one cell, remember this line, listen this line carefully. Any one cell of nucellus, not all cell, not each and every cell. What happened in microsporogenesis? Each and every cell of sporogenous tissue undergo differentiation to produce pollen mother cell. But in this case, any one cell of nucellus undergo differentiation to produce megaspore mother cell. To produce megaspore mother cell. Clear? So this orange color structure indicate megaspore mother cell. Megaspore mother cell, which is also diploid. Remember, surrounding this megaspore mother cell, the other cell of nucellus is still present, but I am not drawing it because it creates more confusion to understand this particular topic. Clear? So I am keeping it very safe and very simple. So in next stage, yes, before enter into the next stage, as we know, differentiation, that means to obtain a particular function. That means any one cell of nucellus now has a particular function, and that's why it is known as megaspore mother cell. Full form: megaspore mother cell. For your clarification, in next step, this megaspore mother cell undergo meiosis. As we know, at the end of meiosis, 4 daughter cell produced. Each and every daughter cell has half the number of chromosome compared to parent cell. So for this process, MMC or megaspore mother cell is parent cell, which is diploid. That means after meiosis, in this structure, total 4 cell produced, 1, 2, 3 and 4. Clear? And each and every cells are haploid because they are produced through meiosis, and meiosis always result into the half of the number of chromosome, and this all 4 cell now known as megaspore. Clear? Now next, 3 megaspore out of 4, 3 megaspore are going to degenerate. I am writing it here. 3 megaspore degenerate. That means out of 4, 3 megaspore degenerate, only 1 megaspore remain alive. So I am drawing it here. Out of 4, 3 megaspore are going to degenerate and 1 megaspore remain alive. And now this single megaspore is known as functional megaspore. This one is functional megaspore. And this functional megaspore is also haploid. Now let's go through the process and understand what is megasporogenesis. See, I already told you, megaspore, we are talking about a female reproductive cell. Genesis, that means production. Simple word, production of megaspore. At the end of this process, functional megaspore is produced. Clear? So what is the definition of megasporogenesis? The fact or the simple definition is production of megaspore or formation of megaspore from megaspore mother cell. Formation of megaspore from megaspore mother cell through meiosis. Through meiosis is called megasporogenesis. So according to your NCERT, this process is called megasporogenesis. Clear? Now, if you recall the structure of megasporogenesis, see what happened. Out of 4, only one megaspore remain functional and 3 megaspore are going to degenerate. That means in future the female gamete is going to produce from this 1 megaspore, and if female gamete produced from this 1 megaspore or only from 1 megaspore, this type of development is called monosporic development. Mono, that means 1. Clear? So here we have only 1 functional megaspore. So this kind of development is known as monosporic development. Clear? So let's move forward. After getting a functional megaspore, this one is functional megaspore. Let's draw it into the center. Clear? Functional megaspore. Now what happened? This functional megaspore undergo mitosis. First mitosis. What happened at the end of mitosis? 2 daughter cell produced and the number of chromosome remains same. That means if functional megaspore is haploid, so as a result of mitosis, the producing new cell are also haploid because ploidy remains same, number of chromosome remains same. Clear? But one complicated thing is that there is not only mitosis. There is process called karyokinesis. If you remember the chapter number 10 of standard 11, there are 2 processes, karyokinesis and cytokinesis. What happened in this process? Suppose this one is cell. If I will say this cell undergo karyokinesis, if this cell undergo karyokinesis, that means only nucleus of the cell is going to divide. Only a nucleus of the cell is going to divide. And if I will say cytokinesis, that means the cytoplasm present in this cell, it is going to divide equally into a 2 daughter cell. That means now we have 2 daughter cell. So as a result of karyokinesis, nucleus divide Karyo, that means nucleus. Kinesis, that means division. Simple logic. Cyto, that means cytoplasm. Kinesis, that means division. In cytokinesis, cytoplasm of cell is going to divide, that means cell divide into a two daughter cell. In karyokinesis, only and only nucleus is going to divide. Clear? So this kind of logic is applicable in this process. So first mitosis, but only and only karyokinesis. As a result of karyokinesis, only and only nucleus, this is nucleus present inside the functional megaspore. Only nucleus is going to divide, not the whole megaspore, not the cell. Only nucleus is going to divide. Clear? Let's see. Why I draw it from the orange chalk? Because now I am indicating only and only this megaspore, not the cell of nucellus, because we don't need in this diagram, we don't need it. We need only and only megaspore. So I zoom megaspore for you to understand what happened when the nucleus is divided. Clear? So this one is nucleus, this one is megaspore cell, and this one is nucleus present inside the megaspore cell. But after mitosis, what happened? This single nucleated megaspore, now it become binucleated. Now it become binucleated. So we get binucleated megaspore. Binucleated megaspore. Actually, this labeling is not important for your board level, so we are removing it. If you want to remember it, you can, but it is not that much important. Next, same process, second time, second mitosis, but only and only karyokinesis, same process. As a result of karyokinesis, this nucleus divide into a two more nuclei, and this nuclei also divide into a two more nuclei. So as a result, now we have total 4 nuclei, 2 from this nucleus, 2 from this nucleus. Clear? Next, the same process will take place third time, third mitosis. Clear? Again, but only and only karyokinesis. What happened? This nucleus will produce 2 nuclei, this one will produce 2 nuclei, 2 nuclei, 2 nuclei. So total at the end of 3 mitosis, we have 1, 2, 3, 4, 5, 6, 7, 8 nuclei. In this cell, in single cell, we have 8 nuclei. Now what happened? See, this all 8 nuclei rearrange themselves into a particular manner. See what happened. These 8 nuclei arrange themselves in a particular order or particular manner. From these 8 nuclei, 3 nuclei are come at the base, 3 nuclei are come at the tip or top part, and 2 nuclei remain in center. It's arranged like this. And now what happened? After this kind of arrangement, actually cytokinesis will take place. Now cytokinesis will take place. Cytokinesis take place in this structure, and as a result of cytokinesis, the cytoplasm present inside this cell is going to divide, and now we have different kind of cells. See, this cell, this particular portion undergo cytokinesis, they produce 3 cells. Clear? This particular portion undergo cytokinesis, it produce 3 different cells. Done? And in center, there is only 1 cell is produced like this. So, if you count it carefully, 1, 2, 3, 4, 5, 6 and 7. So we have 7 cell, and now let's count the number of nucleus. 1, 2, 3, 4, 5, 6, 7 and 8. 8 nucleus. 8 nucleus. So at the end of this process, we got 7 celled and 8 nucleus structure called mature embryo sac. Mature embryo sac. Clear? So this structure represents the mature embryo sac, which has 7 cell and 8 nucleus structure. Clear? Let's revise it from here. Again, what happened? The cell of chrysalis present surrounding this functional megaspore. This particular structure, this megaspore, undergo mitosis, but only and only karyokinesis will take place. As a result of karyokinesis, only nucleus of the cell is going to divide, not the whole cell is going to divide. So at the end, the nucleus of this cell, this whole cell is megaspore. We are ignoring the nucellus which is present surrounding this cell. Clear? So this one is only and only megaspore. Clear? So now megaspore contain 2 nucleus. After 2 mitosis, it contains 4 nucleus. After third mitosis, it contains 8 nucleus. Now these 8 nucleus arrange themselves like this. 3 nucleus at the base, 3 nucleus at the tip. Clear? And 2 nucleus present in the central cell, centrally located cell, large cell. So at the end of this structure, we have 1, 2, 3, 4, 5, 6, 7, 7 cell, and 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei structure called mature embryo sac. So now we are going to understand each and every cell of embryo sac and the labeling of this embryo sac in the next topic. So let's start with the structure of mature embryo sac. Let's see. Clear? As we know, now 3 nuclei arranged themselves in base part, 3 at the top and 2 in the center. After cytokinesis, nuclei get their cytoplasm, their cell wall and all other structure like this. And in center there is large cell is present. Clear? So now let's label these 3 cells present at the base. Out of 3 cells, these 2 cells, this one and this one, this cell and this cell, known as synergids. Synergids. Clear? The cell present in the center called egg cell. And this egg cell carry a particular nucleus, and the particular egg cell is known as female gamete. So from this 7 cell, only 1 cell represent the female gamete, which is egg cell. Clear? This large cell present in the center called central cell. Central cell. And 2 nuclei present in this central cell known as polar nuclei. Known as polar nuclei. And the 3 cell present at the top, all 3 cell called antipodal cell. Antipodal cell. If you observe this structure, the finger-like projection present inside the synergies at the base of synergids called filiform apparatus. Called filiform apparatus. Filiform apparatus. Clear? Now if you remember the diagram of ovule, just for clarification, so it is like this. We are understanding this structure, embryo sac. That means still surrounding this embryo sac there is cell of nucellus. So cell of nucellus is still present surrounding the embryo sac. At last we are drawing it a very large diagram, in a large diagram, so you can clearly understand actually where exactly embryo sac presented. But right now this one or these are the structure of embryo sac. What is the function of polar nuclei? Antipodal cell, synergids, egg cell and filiform apparatus we are going to discuss in next topic, or most probably in the fertilization topic, but right now we need to understand only up to this. Clear? So this structure is called the mature embryo sac. Mature embryo sac which is haploid. That means synergids are haploid, egg cell are haploid, this polar nuclei, that means centrally located cell are haploid, and antipodals are also haploid. Now let's take a look of ovule with the mature embryo sac, how it's look like. Clear? And yes, remember the particular number of cell and number of nucleus: 1 2 3 4 5 6 7 8 nucleus, 1 2 3 4 5 6 7 cell. Clear? Now let's see the structure of ovule in large view, so you can understand how this embryo sac present. No need to draw the funicle and hilum. I am drawing only a body of ovule. See, in this portion the nucellus is present, and in the center, that means here, embryo sac is present. Three cell at the base, 3 cell at the top, and 2 nuclei in the center, in the centrally located cell. And the cell of nucellus present in this region. Clear? These are the cell of nucellus. So for fertilization, it's necessary to provide nutrition to egg cell. Why? Because if egg cell is living, then only it's help in reproduction, help in fertilization. But if egg cell is dead due to lack of nutrition, then it is not able to do fertilization, not able to do reproduction. So now, what is the function of nucellus or cell of nucellus? It provides nutrition to embryo sac, it provides nutrition to developing embryo sac. Clear? So the function of nucellus is most important. Now one more thing, what happened? The end at which these particular integuments are absent called micropyle end, and the end where these particular integuments are present called chalazal end. So M and C are short form. M is for micropyle and C is for chalazal. So now we can clearly say that the synergids and egg cell present at micropyle end and 3 antipodal present at chalazal end. This particular end is most important for MCQ perspective. MCQ is continuously asked like this. For example, this diagram is given to you in MCQ and they are asked, identified at which end synergids are present. Clear? So we need to identify synergids present at M end and antipodal present at C end. Clear? Thank you.